we've been able to really sequence organisms at a high rate that's actually been affordable. And it's also helped identify organisms that we could not culture before. So back in the 90s, you could really only study a bacteria that you could grow in the lab, do biology on, and you could sequence it, but it was kind of tedious. So this ability to sequence anything in a sample has really helped identify a lot of organisms that we were not aware of before. And this project also helped develop tools for not only creating these different libraries for whole genome sequencing, 16S, but also analysis, so creating databases. Because you can only, when you get the sequence data, you have to align it to something. But you're only going to really know what's there if you have a good database that has all the information and the biology of an organism. So what they did was mainly use 16S ribosomal RNA sequencing and started to understand general communities and seeing that actually our different body sites, even the side of your nose is different from what's going on your chin. Everything's a little bit different. And using whole genome sequencing we went from about 150 bacterial genomes to well over 1,500. I don't know where it is now, but we have a whole lot. And we're actually in the process of still doing whole genome sequencing on a lot of bacteria in my own lab. And we've really started to understand what's normal. And I'm not going to go into the details of this diagram, but you can just see the different colors represent different phyla of bacteria. So your GI tract is going to be very different from what's going on, everywhere else. So it's just really, really distinct communities. Another project that we weren't part of, but is really important, I wanted to mention, is the American Gut Project. So Rob Knight and his collaborators at the University of Colorado wanted to just characterize the microbiota of everyone, essentially. So you can pay $99, send it in a stool sample, and they will tell you what's there. So you can actually still do this. So you can Google it and send off a sample, and they will tell you what's living within you, which is pretty cool. And they actually also ask you questions about how does diet influence your microbiome, antibiotic use, even age, and anyone can participate. So what they did first was compare their data to what the Human Microbiome Project got. So their data is the dark red, dark blue, and orange compared to Human Microbiome Project fecal sequences, oral, and skin. And you can see that in the stool, everyone kind of clusters here, and this is the skin, and then the oral cavity. So just those three body sites differ a lot, and no matter who's kind of doing the sampling, you get the same general trends in terms of different phyla of bacteria. If you look at just kind of the average human, red represents the firmicutes, orange is Bacteroides. Proteobacteria make up about 8 to 10% of what's living within you. And if you compare gender, so female to male, it's not really different. Even diet doesn't have a huge impact. You might shift your microbiome a little bit if you go on, say, a paleo diet versus just kind of someone who eats whatever they want. So you can actually make shifts a little bit in your microbiome, but female, your diet isn't really going to influence it too much. What does influence it is antibiotic use. So when you're first on antibiotics, your diversity goes down. If you've been on in the last month, you have a huge kind of bloom of proteobacteria within your gut. Now, your firmicutes, which is red, and Bacteroides, which is in the orange, those are your two main phyla that live within your gut. So they alone make up over 90% of what's there. Firmicutes are gram-positive, they're Clostridia, there's all sorts of different species. Your Bacteroides are gram-negative, tend to be highly anaerobic, and they're really important in digestion. So a whole lot of Bacteroides and Firmicutes is really good for you. However, when you get a bloom of proteobacteria, that can cause problems. Those are the guys, E. coli, Klebsiella, those are the ones kind of like your normal players that cause UTIs, things like that. So as long as they stay in your gut, they might not cause too much trouble, but they get to the wrong spot and they can cause lots of trouble. However, the gut microbiome will kind of return to normal after time. However, you run into issues if you're really on chronic antibiotics for a long time, can really cause issues. Because once you lose a lot of diversity in your gut, it's really hard to get it back. And the microbiome can also change with age. So babies are pretty diverse, and it kind of stabilizes through time, and then once you're older, you see a lower diversity. So just a general overview of kind of what we have in general. At the phylum level, everyone is very similar. So high numbers of Bacteroides and Firmicutes living within your gut. However, when you get down to a species level, everyone's actually very different. So my microbiome is going to be very different from anyone else out there in this room. And that's at the species level. However, if you look at the overall function of your microbiome, it's pretty similar. You know, digesting food, metabolism, and essentially, you know, the core genes are the same. However, having high diversity, no matter if it's your skin, your gut, that's really key to good health. It's when we see kind of a lower diversity that you start to notice problems. And we're starting to understand kind of, okay, what bacteria are good, what bacteria are bad, because I've just thrown a whole bunch of names at you in terms of phyla, but I can't really tell you, you know, too much about what they're really doing, who's the really good guys, who's the really bad guys, because we're just starting to understand that. And this is where the Human Microbiome Project Phase 2 comes into play, and they've done kind of a consortium that's more of an integrative human microbiome project, and our lab is playing a major role in that. And the focus is getting a whole lot of collaborators from not only all over the country, but even having collaborators all over the world, seeing how the microbiome really plays a role in health and disease. And so what they're doing is taking 3 main cohorts of people. This includes pregnant women and their babies, people with inflammatory bowel disease, and people who are pre-diabetic. And what they're doing is taking these 3 main groups of people and doing longitudinal data sets. So looking how the microbiome changes over time, and I will go into over the next few slides about how they're doing that for these 3 different cohorts. And doing very detailed, deep analysis on all 3 different cohorts to really understand what's good, what's bad, you know, what seems to be really correlated to certain bowel diseases. Because, like I said, we still don't really understand it. You can find trends, but we're really just starting to understand what's going on. It's a very new field. So I'm just going to talk about the first project, which is focused on pregnant women. So during the course of pregnancy, the gut and the vaginal microbiome of a woman changes, and it actually gets less diverse. And as I said, lower diversity is actually usually not good for you. So in the gut, you see increased proteobacteria, which I mentioned are not really good for you, increased actinobacteria. And in the vagina, you see more lactobacillus, clostridium. And if you just look at, this is just representing bacterial species. So when you're not pregnant, the more color you see, essentially, the more diversity. So, and if you look at someone who's pregnant, they actually have lower diversity. They have a lot more lactobacillus species growing within the vagina. And we don't really understand what that means. So that's one focus, is really looking at that and understanding what that means. Because a lot of women who experience preterm birth, about 25 to 40% of it is due to essentially a microbial infection. So either they're getting an infection that's coming up from the vagina or descending from the mouth, and that certain species are being correlated to preterm birth. So it could be Sneathia amni, Gardnerella vaginalis. Gardnerella is actually associated with, oh God, what is it? It doesn't matter, something bad. Trichomonas and vaginalis is another one. And they actually don't really know how these 3 could really be causing an issue. There's some indications so far, but you know, the other thing is to really understand the biology of these 3 microorganisms. The other concern that they have is that babies are not born with a microbiome. So babies come out, you know, they're completely sterile. So when a woman goes through normal birth, the baby is actually first exposed to bacterial communities coming out of the birth canal. And at first they'll see a lot of lactobacillus in the gut, that kind of thing, because it's coming out through the vagina. So, and then the microbiome will actually transition to a normal, diverse, healthy state over time. However, women who get C-sections, babies don't get that exposure to the birth canal, right? They're just kind of taken right out. So essentially they're exposed to the mom's skin, even things in the hospital. And as we all know, hospitals are breeding grounds for bad things. So babies born by C-sections don't get that exposure right away to a very diverse microbiome. And they're actually finding correlations to asthma and allergies and other issues just from having a C-section.